All right, so next up we have Brian Ott. He earned his bachelor's degree in marine biology from the College of Charleston and a master's degree in biological sciences from the University of Alabama before joining the USDA ARS Warm Water Aquaculture Research Unit in Stoneville, Mississippi as a fish biologist. Brian is currently a PhD candidate with the Department of Wildlife, Fisheries, and Aquaculture at the Mississippi State University College of Forest Resources under the guidance of Dr. Peter Allen. His primary research focus at WARU has been improving production efficiency in catfish aquaculture through oxygen and water quality management in channel blue hybrid catfish systems. So you can take it away, Brian.
All right, thank you very much. Dr. Griffin asked me to give this presentation about a week ago, and so to fill in for someone. So I'm unfortunately the B team, so you're stuck with me. I also any work on disease, so this is all disease-free in this presentation. He just thought some of the stuff I worked on is kind of interesting. I do have one slide of dead fish, so I hope that'll bring you guys back home. One final disclaimer. When I was asked to give this, I didn't realize that my wife was going back to work to teach this week. So I have two young children upstairs, and they're being tended to by Ghostbusters. So hopefully Bill Murray will keep them fully engaged for the next 20 minutes here. Otherwise, I apologize. So this is where we work. This is Stoneville, Mississippi. And this is a sort of a aerial shot of a lot of our research ponds. And it would be cool if we worked in a sea of blue out here, but unfortunately, or for better or for worse, it's a sea of green. And so you can see out there all these different ponds. There's some ponds in the middle there that don't have any water at all. Of course, there's a bunch of dirt there, but everything else is varying shades of green all the way up to a light brown. And what you're looking at is basically phytoplankton. Catfish ponds are largely regarded as a phytoplankton or autotrophic-based systems. And so what that means is that you have, the majority of the nitrogenous waste that's produced from the catfish or the feed is eliminated through phytoplankton. And so what we have in these ponds is a whole bunch of chlorophytes and cyanobacteria, and they basically dominate those ponds. And they're extraordinarily important. They really are kind of the stars of the show out there. If you consider a catfish pond kind of like a factory, these phytoplankton are actually like the factory workers. They're out there getting it done. They're making everything happen out there, and they control many aspects of the water quality within a catfish pond. So oxygen. At a sort of in a static state, the oxygen in our ponds is about 8 milligrams per liter. That gets higher in the wintertime when the water is colder, and it also gets lower in the summertime as the water gets hotter. And so, but if you kind of take an average, it stays at about 8 milligrams per liter. But it doesn't just stay like that. Throughout the course of the day, that DO goes up and down. And basically, it's in response to sunlight and, of course, photosynthesis. So as the sun comes up over the horizon, some of these phytoplankton are now able to undergo photosynthesis, and you get this production of oxygen, or a net production of oxygen. Then when the sun goes down, now you get this decrease in dissolved oxygen concentration. So basically, sun comes up, photosynthesis starts, and you get a net increase in oxygen. But meanwhile, when the sun goes over the horizon, you start to get that slowdown of oxygen or net oxygen production, and it results in a decrease in the oxygen concentration in the pond. So where does that oxygen go? Well, most of that is going to the phytoplankton. They consume up to 60% of that. The sediment and the bacteria in the sediment consume approximately 20% of the oxygen. And finally, a fish at 6,000 pounds per acre consume about 20% of the oxygen. Now, these numbers were from a few years ago. And so since then, we've increased the production intensity. So these numbers probably are not super reflective of the sort of the more common, modern day ponds, but that's where they were when these values were calculated. But during photosynthesis, there's a lot more going on simply than like the changing of oxygen concentration. You also have carbon dioxide that increases. Basically, it's the opposite of oxygen. So, when they're producing oxygen, they're consuming CO2 at nighttime. There is no more of that CO2 consumption, but you have the fish producing all the CO2, and so now you get an increase in CO2. In response to that change in CO2 concentration, you get a change in pH. And so, basically that kind of goes opposite the CO2, kind of mirrors out of oxygen. And so you get this daily pattern where it's very kind of dynamic in the changes of oxygen, CO2, and then therefore pH. Now, pH changes in response to CO2 concentration, but it's limited kind of basically by the of the alkalinity in the pond. So in the ponds in the Mississippi Delta where we are, the very high alkalinity, so the pH does not change as much where compared to, say, East Mississippi, West Alabama, the alkalinity is very low. In those ponds, we get much greater swings of pH. So it's important if you're doing, one of your studies, if you go take a water sample from a pond at any given moment, it's simply a snapshot of time. And if you go out there like an hour later, maybe, you actually find some pretty big changes in regards to these parameters. So something to think about as you're kind of designing experiments or looking out there at your ponds. But to go back to the oxygen, this is kind of, most bodies of water, except for things like, except for like oligotrophic lakes, are going to have some changes in oxygen concentration in response to photosynthesis. But with us, when we feed our fish, we're effectively fertilizing these ponds. And so what you get are these really dramatic swings where you get to all the way up to maybe even 250% saturation. But if you notice, the curve gets all the way down to zero there. Without supplemental aeration in the heat of the summer, our ponds would basically, suffocate our fish every single night. And that's kind of what that looks like. That was a small 1/4 acre pond. I fixed an aerator, forgot to turn the aerator back on afterwards. And so, of course, that night, here you go. And so not good. But so all these ponds, we were actually actively feeding these fish have some type of supplementary aeration. For us, that looks like this. This is a standard paddleware aerator. For you people up in Minnesota that haven't seen them, maybe that's what it looks like. That's anywhere from 15 to 20 feet wide. It runs basically at nighttime as the oxygen gets low. And so we designed, over the course of 10 to 15 years, we designed a number of experiments to kind of determine the effects of minimum oxygen concentration on fish appetite. And so we completed these studies with channel catfish, blue catfish, and hybrid catfish, the hybrid of the two of those. And basically what we did, and I can show you, this is the results from one of the studies. All of the studies we did largely look the same as this. If you look at this, what we did is we controlled the minimum oxygen concentration of these ponds at nighttime through aeration. And so our controls had the oxygen concentration reach like 3.5 to 4 at nighttime, whereas the experimental group, or I guess maybe that is more sort of the control, got all the way down to 1.5 milligrams per liter. Now, to give some perspective, historically in the catfish industry, they would let their oxygen get all the way down below 1 even. So these fish can survive very low oxygen concentration. And that was kind of how they managed their ponds. I once met a catfish farmer, and he told me he had the best aerator guy in the business because he rarely used his aerators. He let his fish get down to points every night, points being under 1, and he rarely had to use his aerators. And he thought that was a huge success, and he was bragging about it. But apparently he missed the last 10 years of our presentations, but that's okay. So anyways, if we go through this, this is kind of how the experiment unfolded. You see April, there's not much feed going in, the water's kind of cold, so the oxygen is very similar between our two groups. But over time, as you start to feed the fish and the fish grow larger, you're fertilizing that system more, the phytoplankton population increases, and now you start to get these sort of dramatic drops in oxygen concentration. And so June through September is when the majority of your feed goes in, and that's where we held the lowest group at 1.5 and the highs at 3.5 to 4. And so how that's reflected in the feeding looks like this. April, before there was a change in the oxygen, they were the same. But after that, there was an incremental disparity between how much feed the control groups, or sorry, the low oxygen groups and the high oxygen groups consumed. And so what you see over time is this reduction in appetite. And it looks like their appetite is getting less in the low oxygen group, but really what you're looking at is a decrease in fish size. So those fish, ever since May, are growing a little bit slower. And so, of course, they eat a little bit less, even if they were eating the same proportion of biomass, which they aren't anyways. And so here's the production data. This is what this looks like at the end of the season. Well, survival in all of these years, unless something bizarre happened, is the same. There is no impact on survival with low oxygen or high oxygen. Same with FCR. With the ranges of oxygen concentration we're looking at, there's largely no change in FCR. And this is, especially physiologically. Now, FCR, one component of that is, the fish physiology. The other component of that is actual fish management in terms of fish dying out there. And so if, your fish grow slow enough and you have to grow them an extra month or 2 or 3 or 4, it's possible then with some added mortalities that you would artificially increase, which is worsen your FCR. Of course, if your fish eat less, they don't grow as much. Then, of course, if your fish weigh less and there's the same survival, the production is less. So basically what we found is as the oxygen decreases, they eat less feed. And so if you compile all these studies into one graph, you kind of start to see what that all looks like in totality. That up here on the right side, those are the control ponds. And then as you, and so the x-axis is the minimum dissolved oxygen concentration. As you move left to these various, each dot represents an individual study. And as you move left, those fish eat less feed. And so that's kind of what we found. So basically our recommendation is you want to keep your minimum dissolved oxygen concentration above 3 milligrams per liter. Now, you say, well, what about more than that? The problem is that as you increase, as you target a higher dissolved oxygen concentration, it's approximately a 14% reduction in aerator efficiency as you go up 1 milligram per liter. And so your aerators progressively get worse the higher you want to maintain your oxygen. They get progressively better the lower you want to maintain them. So this is kind of like, at a reasonable stocking density with a paddle wheel aerator, where you're relying on diffusion to oxygenate the water. This is sort of like a practical limit to that. So this kind of begs the question going forward then, is like, what are the limits of these earthen systems? And the common thought is that it's going to be ammonia or some type of nitrogenous waste. So either the excess feed or the fish are excreting different types of ammonia. And so that, at some point that becomes toxic, it suppresses appetite and maybe even kills your fish. So we designed a project a couple of years ago where we looked at, we wanted to basically overwhelm the system with ammonia and nitrogenous waste, basically in an effort to get to the point to where the fish are stop eating as well or they die. And so with the constraints of space we had 6 ponds. We added a single density for each pond ranging from 6,000 head of the acre up to 36,000 head of the acre, which is an extraordinary amount of fish. 6 to 15,000 head of the acre. And so basically those 2 left dots is where the industry works. We worked much, much higher than that for this one particular study. It turned out pretty well this year. This is our feeding graph. For each individual pond, you can see the feed we fed on average that month. These phytoplankton ponds, as you fertilize the ponds, those phytoplankton populations increase. But at some point, they start to shade each other out, and there's a practical limit to how much phytoplankton you can stick in a pond. So that should mean that there is a limit to how much nitrogen can go in, and then how much nitrogen they can take out. And so that limit basically, lives around here. And it's kind of 150 to 200 pounds an acre. That is sort of the theoretical limit based on the theoretical limit of the phytoplankton population. So what happened with the water quality here? So you saw the feeding, what happened. We fed a whole bunch of feed. If you look at the chlorophyll A concentrations of these different ponds, it basically, this is a flat line, statistically speaking, and it kind of reflects, the maximum chlorophyll A concentration you'd expect in these ponds, anywhere from 400 to 600 micrograms per liter, and that's pretty much where we hit, and we stayed there once we hit the maximum. But this doesn't quite tell the whole story. If you look at an individual pond, this is our highest density pond, and that's sort of the theoretical maximum. Every so often, it starts to actually get well above that, and it's followed almost immediately by a crash back into the that like sort of expected maximum range. And so I thought that was kind of neat. And again, this goes to show that these ponds are highly dynamic. The phytoplankton population will change with temperature and with feeding rate. And so there's a lot, they're constantly evolving and changing. And I think this is reflective of that. And again, so, any given moment you go out there and look at that pond, it's just a snapshot in time. And you really need to start to look at like maybe a broader time range to really get a handle of what's going on there. So if you look at ammonia, we went all the way up to this super extraordinary amount of feed, and we had no significant increase in ammonia. Same with unionized ammonia. So, it's just, it wasn't there. But when you get to nitrite, we had a significant increase, as well as nitrate. Nitrate, we've never really measured before, or most folks don't measure, because there's really no reason to expect that there's an increase in nitrate. And so the reason we have nitrite and nitrate is that we now have an increase in bacterial nitrification. To achieve that, you have to have some sort of spillover of ammonia. If that ammonia is all being consumed by the phytoplankton, there's nothing left to kind of seed your system. I'm sure everyone in this talk has a fish tank, and you have to kind of seed your fish tank for 2 weeks with some feed, or maybe a sacrificial fish to get your bacterial nitrifiers going in your biofilter. Well, the same thing happens here. You need some ammonia spilling over those phytoplankton to actually be caught by those nitrifiers to get that system going. And so I think that's kind of what happens here, is that over time, once you reach that maximum phytoplankton population, start to get some excess ammonia, and that's how we can get these nitrifiers going. And that being said, there's other avenues for nitrogenous waste to leave the system. There are heterotrophic bacteria. I don't know a whole lot about those, but those are little guys that run the bioflocks. You heard of like the shrimp or tilapia bioflocks. They use heterotrophic bacteria. They have to seed that system with carbon. It's basically just like a compost pile in a fish pond, and that's another way to remove nitrogenous waste. Another possible way is actually you can volatilize the ammonia. That's basically degassing your system through aeration or by some means to actually remove ammonia from your pond. So there's a couple possible avenues for ammonia to leave the system. We only measured bacterial nitrification, and so we know that's going on to some degree. But there's others it's possible working at a couple systems simultaneously. So I want to go over some of the physiology stuff that I've worked on, and I'm just briefly going to hit on some of this, because some of this kind of dovetails with some of the work you guys do. And so I developed these hypoxia assays in the lab, and it kind of looks like this on the graph on the left there. We did the control pre-hypoxia period for a couple days. Then we did 12 hours of hypoxia. We sampled them twice during that, and then 12 hours of recovery afterwards. And it's basically all done in tanks like this. And I also wanted to point out that the very first response typically of a catfish to hypoxia is like a behavioral one, which is this. They all crowd around the water source in an attempt to pick up as much oxygen as possible. In a pond, that means they get behind an aerator and they start swimming, and they get in that flume of that aerator, and that's like the most oxygen-rich environment in that pond at that moment. In my tanks, it happened to be my outflow of my water there. Unfortunately, there still wasn't much oxygen going in, but that's where they wanted to go. So I always think that's kind of interesting, and you shouldn't forget that. But if you look at the venous PO2, which is the partial pressure of oxygen in 6 hours, they're significantly lower. 12 hours is even lower than that. And then 18 hours, I'd have to check my paper, but I I think that's, it looks like it's significantly lower. Then by 24 hours, they've fully recovered. So you see this decrease, and I'm sorry, that's the period of which I ran hypoxia in this experiment. So you can see, through a variety of means that they employ to try to maintain their internal, sort of oxygen concentration or partial pressure of oxygen, at some point they just can't quite keep up. And so that's what happens. So to take this even further, I think this is sort of interesting. We did another series of experiments where we did this sort of periods of cyclical hypoxia, kind of mimic what happens in the pond. So in the pond, you have 12 hours of high oxygen followed by approximately 12 hours of low oxygen. It's a little more nuanced than that, but that's kind of the idea. And I sort of replicated that in the lab. So over the course of 5 days, high, low oxygen. I coupled that with a series of tanks that maintained just pure chronic hypoxia. And so they were never removed from the hypoxia over the course of 5 days. And it's pretty amazing. If you look again at the venous PO2, the gray fish, the solid blue bar is the chronic hypoxia, and then of course the ones that went those daily routine there. And you can see they bounce right from the control group down to the chronic group, back to the control group, and so forth and so forth. So it's almost kind of hard to imagine this is going on for like 110 days straight in a catfish farm, where these fish are experiencing these short-term bouts of sometimes extreme hypoxia. Another thing we measured lots of stuff. Here's a couple of the highlights. One was hematocrit, and I know one of your first seminar speakers, Ms. Gunn, she measured hematocrit on these fish that were infected by Bulbophorus, and it kind of destroyed the hematocrit. And I think it's interesting if you can kind of potentially conceptualize the interplay between this, hematocrit increasing during periods of hypoxia and then decreasing during periods of normoxia, coupled with a infection that may severely impair that. I think that would be potentially interesting. Running out of time here, I just wanted to show you some very last piece of work. We did some RNA sequencing on the hypothalamus. That at the end of those 4 steps, a bunch of data on that, but just a brief overview is that if you look in the hypothalamus there, you have 6 to 12 hours during the period of hypoxia where you have a pretty dramatic upregulation of a number of genes. But once they get back into that recovery phase, a lot of those genes that have been differentially upregulated have now sort of stopped and have returned back to control values. I think that's pretty interesting. And you get, we can talk more about this some of the time. So anyways, in conclusion, I just wanted to point out that catfish ponds are extraordinarily complex, dynamic environments. There's a lot of stuff going in there on a daily, potentially even hourly basis. If you're raising catfish, you've got to keep your ponds above 3 milligrams per liter in regards to dissolved oxygen concentration. It appears that bacterial nitrification increases with ammonia concentration. Once you get to a certain limit of the phytoplankton population, you start to get this increase in bacterial nitrification. And finally, life is tough in the pond. There's lots of, there's a lot of stuff going on that these fish have to cope with, and I think that's pretty amazing. So anyways, if you have any questions, I'm happy to take them. I'm sure you have my number or email from the announcement, so please don't hesitate to contact me. Thank you.
All right, thank you very much, Brian. Great presentation. We got plenty of time for questions if anybody has them. While they're thinking, Brian, I've got one for you. So I'm impressed. Those catfish seem awfully resilient to be able to bounce back and forth between the oxygen levels, just physiologically. Is that something you'd expect in other fish too, or is this sort of resilience and tolerance for low DO pretty unique to catfish?
So I think the answer to that is I don't know for sure. My guess is that there's certain species that are clearly adapted for more like athletic, thinking like a rainbow trout. They're sort of evolved like an athletic lifestyle in a very consistent environment, very consistent oxygen concentration. It's going to be very high. And so if you remember back to your fish physiology courses, they have these, you know, the P50 curves. They can offload oxygen really fast. So they can, you know, swim really quick. Whereas a catfish is different. You know, they're evolved to be in these potentially really low oxygen low oxygen environments. And so, they're obviously, able to cope with this better than, say, a rainbow trout. So I suspect you're going to have like a whole wide spectrum of fish where you go like rainbow trout on one side that cannot deal with this as well, all the way down to like a bullhead that can, like, no problem at all for that. And so, so within the, that being said, could a rainbow trout, deal with this, like within the limits of their physiology, maybe going from 100% saturation to 75? I would think so, but, I've never looked at that. I'm not sure. And a lot of these diurnal daily like fluctuations in oxygen, it's not super common. It's common that it happens, but not at the range which these catfish are experiencing. So not a ton of work out there looking at some of these like daily repeated bouts of hypoxia.
Cool.
Hey, Brian, this is Jan. That was a great presentation. Thanks. And I think that you definitely are talking about fish diseases as well. So just to commend you on that, I think that this is the cycle of these ponds, the DO fluctuations, and all these differences. That could be really an underlying factor for fish diseases, whether it's catfish ponds or it's wild fish. So I think that this is like a really important principle for fish disease folks to keep in mind, because these are some of the stressors that lead to other diseases that are out there. But along that line of thought, something like proliferative gill disease, do you see like highest mortalities? I mean, that's a common catfish gill pathogen, and it might not be a safe assumption to think that it's hypoxia that those fish are dying of, but presumably they're less able to cope with low dissolved oxygen. So do you see these ponds that have high phytoplankton or that have these largest fluctuations? Are these the ponds that are also losing the most fish to proliferative gill disease? Is anyone really looking at that?
So that would be a good question for Dr. Griffin. And I'm sure someone's mentioned this, but so as I mentioned briefly, the very first response to these fish when they experience hypoxia, they swim up behind the aerators. And so they spend all night long swimming behind the aerators. And one of the management techniques for the PGD is that they'll have to sometimes run the aerators during the daytime, because these fish are basically acting like they're undergoing like hypoxia stress. And so if you walked out to a pond and go, oh my gosh, these fish have no oxygen in the pond. Well, the oxygen's fine. They're just loaded up with PGD. I've never had to deal with that personally, but I know that's one of the things they have to do is they may even run the aerators during the daytime. And so how that interacts with, so basically, the larger your phytoplankton population, the more feed going in, the broader these swings of oxygen concentration are going to be. And so, it's going to be a more protracted period of hypoxia. And so instead of maybe being like an hour or two of really short, like intense hypoxia, it might be 5 or 6 hours. And so you have that, to couple with. So as you increase the phytoplankton, you're almost naturally always going to get lower oxygen for a longer period of time. And so if you're dealing with a fish that has some sort of, disease, or sorry, a gill, infection, I think, of course, that's going to, exacerbate that problem.
Sure. Yeah, that makes sense. Great, thanks. And it seems like these producers, if they do have a lot of problem with proliferative gill disease, maybe that's another recommendation you can give them to try to avoid those swings and to watch your phytoplankton and all that stuff as well.
Yeah, it's really hard. Caroline mentioned copper. Some folks add copper to kind of like moderate their phytoplankton populations, how to keep it from getting really high. But, if you want to feed your fish and you want to feed them well and you want to grow a lot of fish, you're going to put a lot of fertilizer in the pond. You can't get away from that. That's just one of the things you have to work around. And that's why we have giant aerators.
Yep, sure. Cool. Thanks, Brian.
Hello, Brian.
Hi.
This is Ellen Lark, and I wanted to just I hope it may end up being a helpful comment. If you have in your phytoplankton a sizable population of cyanobacteria, they may be adding to your nitrification because they fix atmospheric nitrogen.
Yeah, some of these ponds have very large populations of cyanobacteria. You broke up, so I kind of missed your part of your question, but I think I got it all.
Well, it was more a comment than a question, is that you have cyanobacteria that are able to fix nitrogen.
Mm-hmm.
So they may be adding to your nitrification.
Yeah, it'd be something to think about for sure.
It's not just sedimentary bacteria doing that.
Right. And the systems that we designed, that we worked in here, it's extraordinarily high amounts of aeration, 3 to 4 times the size of the industry average, not size, the amount of aeration. And so what happens is we get much higher turbidity than you may expect. And so we have, relatively high suspended solids in these ponds. And we think that kind of maybe offers sites for these bacteria to colonize when they're kind of floating around the ponds. And so it's not, I think, our nitrification measurements are all taken in the water column. We didn't do anything with the sediment. And so we know that there's enough stuff floating around that is kind of, we're getting like these extra sites for bacterial colonization.
Anyway, very interesting and talk, and I enjoyed it a lot.
Thank you.
Any other questions out there?
I have one. This is Alex Primus. Just thinking about nitrate in these ponds and how the nitrate gets out of the ponds. I imagine some of it might be used up by the phytoplankton, but there are probably some bacteria that you mentioned, some of these heterotrophs that are undergoing denitrification.
Right.
I don't know if it's well known how many of those heterotrophs that are denitrifying are functioning in aerobic versus anaerobic conditions. But I just thought I'd ask if you know anything about that.
Yeah, I don't know a whole lot about the denitrifiers, but yeah, you're right. So these nitrate can get repackaged back into phytoplankton. So that's a lot of what you see is maybe this ammonia in the different nitrogenous species basically gets repackaged into phytoplankton. Phytoplankton die and it gets absorbed by something else, a couple of days later. And so you kind of get this recycling of that over and over again. And that could also, I think they're preferentially like ammonia and nitrate as their two, favorite types of nitrogen. And so at least that we have there. And so, yeah, I think you're right. And there's almost certainly some denitrifiers, but I've never measured that. I don't really know how to measure that. And, but to be honest, though, for years we never really cared about nitrate, and I don't think we should care about nitrate at this point because it's just not accumulating that much. We're well below the threshold of possible like toxicity to the fish, and so it's simply not a problem or an issue at this point. But I do think it's a really interesting ecological question. What is the whole makeup, the metagenome of the pond? What's going on inside of there? What does that look like? How does that change with time? And how does that change with feeding rates? I think that's all kind of stuff. That's kind of where we're heading, I think, in the future. I think that's all, sort of interesting stuff. Yeah.
Okay, thanks. Yeah, I guess I come at it from the perspective of working more with people who are working on RAS facilities, right. Sometimes brackish RAS facilities, and obviously nitrates there are can be a bigger issue if you're trying to conserve water. I just wasn't sure how that happened in the natural system as well. But so thanks for your input.
Thank you.
All righty.